Okay, kids, we're going to go over translation and how the ribosome finds the mRNA. We're going to start here, prox, right? We have a small subunit, a large subunit, and then together, this is our friend, the ribosome, that we talked about. Now, the Shine-Dalgarno sequence is a sequence in the mRNA that helps guide the small subunit to find that first AUG. We are not going to memorize the Shine-Dalgarno sequence, but we do know it's the sequence that matters. That's what helps the small guy find, small ribo, find essentially the start, right? Start of translation. All right, let's go. Let's look. We're not going to talk about we're going to talk about it, but we're not going to memorize what proteins help all this because there's so many that probably are not even in our book yet. Okay, here we go. So here is the Shine-Dalgarno sequence just so you can understand it. And we're not going to memorize it, but it is a sequence of the rRNA. 16S, that's the small ribosomal subunit. Oh, in prox. Oh, that's my handwriting so bad. Right, we're not going to memorize it, but this little here is what I'm saying. If we change the sequence, this would no longer do what? Complementary base pair. And complementary base pairing is guiding this interaction, finding, right, that first AUG. So this sequence is part of the small subunit. And so it's that complementary base pairing of rRNA to mRNA. Again, we're still in prox. How do we know? Because it's a Shine-Dalgarno sequence. We don't have that in eukaryotes. Right, here we go. Shine-Dalgarno sequence, first AUG, right, allows it to attach, takes some energy because we're doing work. We're not going to talk about which kind or whether it's GTP or ATP. We have a formulated methionine. That's the guy that comes in for the first AUG. The rest of the AUGs in an mRNA just have regular methionine. Only the first one is formulated with that little F. Okay, the rest of them, remember, you can have methionines all throughout your mRNA. You have to have one near the 5' end, right? Where this is 5', this is 3', 5', that's 3', of course. So we can have more of them. It's just they're regular methionine. Only the guy first is formulated. That's only in prox. Okay. So it gets on there, this complementary base pairing, anti-codon to codon. Remember, anti is always in the tRNA. So tRNA, anti has a T in it. Right, the codon is always in the mRNA. What else? Stuff happens. The big guy, the large ribosomal subunit, jumps on after this is assembled. Again, prox, but the large guy also hops on last in eucs. So remember, small, then large. Well, small, tRNA, right, with the FMET, and then large for prox. Ooh, what about eucs? Right, same idea. We have a 5' untranslated region whether we are proc or euc. We also, they're not showing us here, which is stupid, we have that methylated cap, right? That methylated guanine cap at the 5', and here's our poly A tail, 5' untranslated, start, stop, 3' untranslated, poly A tail, right? That's our fully processed mRNA. No introns, right? We took those out already too. We're not talking about that here, but there's no introns in there. It's just exons. What happens? The proteins attached, right, to interact with that cap binding protein, which finds the cap and enhances the ribosome to the 5' end of the mRNA. I don't really like this picture so much, but let's move on. Yeah, so the small subunit, you know, starts and grabs the cap with cap binding proteins, finds, you know, the first AUG and slides along to get that RNA started. All right, Kozak sequence. This is similar to the Shine-Delgarno. So analogous, not the same, not homologous, analogous, similar function, except it's right around the first HEG. So this G, super important. So this is position 1, 2, 3. This is -1, -2, -3. This is actually R, which is an A or G. So some kind of purine at -3, super important for that Kozak sequence. And again, to get that small subunit. Let's go. Get in there. And so, what is going on? Now we're right back to prokaryotes. Huh, rude. Going. Here we go. Okay, so now we're back. Now we're back to the prokes. Wow. That's a little confusing. Anyways, we're fine. You guys are fine. And so this is, right, here we go. We're sitting right at the first AUG, and we have the EPA sites, and the ribosome reads 5' to 3', so it's going to be moving along the RNA in that direction. And so you can see it's sitting in the P site, the peptide site. And then as it moves, we're going to have another RNA, tRNA pop down and bring in a amino acid. And then, right, we get some action going on right there to help to get that peptide bond. Once that peptide bond is formed, then the ribosome shifts. The AUG is in the exit site, and that guy exits. Ooh, interesting. The peptide site is where the existing tRNA with that protein, and then the A site is the acceptor site, right? So this is the acceptor site. This second one pops down into the acceptor site. We get magic happening here. We make that peptide bond. We shift. The peptide bond is broken from the tRNA and the amino acid to come off of the guy that's in the P site. Methionine attached to the guy that was in the A site here in the P site, and that guy leaves. So again, you should watch the animations for this because it's so much better than trying to do this in a static way. And that's posted to Canvas, of course. Let's see. Do we have this? Now, over. All right. So then here we go. We're somewhere in the middle, right? We're not at the end. So we're during elongation because you can see there's a whole bunch of amino acids. These are each an amino acid. The P site's the one that has that long peptide, right? This is the A site where all the tRNAs came in. But if you run into a stop codon in that A site, it's not a special tRNA. Someone always falls for this. Don't answer special tRNA in an exam or quiz. Special tRNA. No, we're doing this. No, there's no such thing as that. It's a release factor, several release factors. They're proteins. They come in there. They sit right on that mRNA in the way. So another tRNA, right, holding methionine, we'll just say methionine, any old amino acid can't sit down. There's no way we can complementary base pair with that guy. Okay, so we're out. No way. Nothing can come in there. There's no such thing as a special tRNA. It's a release factor. And then again, when that happens, this whole thing just blows apart. So we're just going to bring it. Boom. Done. Right? Explodes translation, and that is termination. Termination. All right. So, all together now, we've got our charged tRNA there, anticodon. We got to find that first AUG, the small guy, the tRNA, the big guy. That's initiation, right? And then elongation, we keep shifting that ribosome moves. The ribosome moves. Somebody goes into the exit site, he leaves. The guy with the existing polypeptide is in the P site, and then the A site's empty, a new guy comes in, blah, 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 until the A site has a stop. That's termination. And then peptide is released and everything blows apart. Boom. Okay, so this is generic. Could go for either proc or euc. Woohoo! What else? Components
here
required
is our
for
list
protein
of things we
synthesis
need
in
to know.
bacteria.
We don't
So what
need
is a
to know-
bacteria?
So the
Proc.
binding of
We are going to know
the
mRNA
amino acid
and
to tRNA.
methionine. These
right,
are
that's
the
amino acyl tRNA synthetase.
This is the ribosomal subunits.
need, yes,
right. This is
ATP
small.
to
You
do
don't have to
that.
know it's 30S.
I'm writing.
This is
Ooh.
large. You don't have to know it's 50S.
We need the small
You don't
subunit,
need to
large
know
subunit, mRNA.
what these are called or what
Formulated
exactly they
methionine.
do.
Oh, we need
So
to know something about
they
these
do
guys.
exist.
That they exist, but not
Right.
exactly what they
And
do
then
or the differences
elongation,
between them.
right? 70.
Right, there's the
S
big guy,
complex,
the total
which
large.
is small and large together. So the real ribosome.
Yeah, see? Look at me. Ooh, the large ribosome.
Yes, the charged tRNAs.
We need the charged tRNAs that were made up here from
elongation
amino acids
factors.
We definitely need elongation factors,
peptidal
but we're not going
transferase.
to
That's
know
the guy.
about which one does
I
what.
knew that.
And then
Sorry.
this guy, right,
Peptidal
makes the
transferase
peptide
is the guy
bond.
that
So
sticks the
amino acid to amino acid
on the
together. Ooh,
tRNA.
I'm lying.
Woo!
I
What
forgot.
the heck?
I forgot.
No,
I
no,
forgot.
no.
Amino.
So
I don't know
that's-
what I wrote. Amino acid to amino acid. So that regular peptide bond, peptidal
shit.
transferase. Again, it's right in the name. And then termination. We do need to know we have release factors. We don't care about 1, 2, and 3, and that they bind to the ribosome when stop codon is reached and terminate translation in the A site. Right, when the stop codon is in the A site. And then boom, it blasts it all apart. Good. And then the direction of transcription, again, in procs, co-transcription translation, because there's no nucleus, right? You're just in a prokaryote. Ooh, here's your phospholipid membrane, right? Oh, maybe you have a cell wall. Peptidic glycan, or maybe you have some flagella. Okay, sorry. So transcription and translation all happening in here. Transcription, translation. There's no processing, remember? And so, of course, we are building. Here's our RNA. That's the first part of the RNA, 5' to 3' from the template strand. And as soon as any of the mRNA comes off, you can immediately hop, right, a ribosome on it. And then you're, right, this guy's jumping on, right, this guy's been on, longer. So the longer, right, and then at some point when this must have been what? Stop codon, right? And then it blasts that whole thing apart. So if you're looking at it, you can see, right, the peptides get longer and longer. So if you're here and you don't know which is the 3' end and the 5' end of an mRNA, and you have a little protein or 3 amino acid protein here on a ribosome here has more, ribosome here has more. Which direction is transcription? Which is the 5' end? Which is the 3' end? This would be the 3' end. This would be the 5' end, right? Because this guy is where translation would start here, so it's had a lot more time. And each ribosome doesn't have to wait till one's completely done to jump on. Polyribosomal. Lots of ribosomes can be on at the same time.
and the difference between procs and eucs in the details of gene expression. So again, this is always a good thing to know. No nucleus in the procs, right? Transcription, translation all together. Nucleus separates from the cytoplasm, so transcription and translation are separated, and we have to do RNA processing first. Cap, splice, tail, maybe some more splicing. And, right, talking about the introns had to have come out, right? The gene is what it is, right? The mRNA is exactly off the DNA. We have to actually do processing first. So primary transcripts are the mRNA for procs. We had to cap, splice, tail. And then, look down here, initiator tRNA, formal methionine, eucs, regular old methionine. This guy has Shine-Dalgarno, this guy has Kozak, and you can have multiple ribosomes at a time for both. Small ribosome binds the first methylated cap, 5' end, and then scans the mRNA to find the ribosome binding site, that first AUG, then we can get that tRNA, then we get the large subunit come in, right? So same small first, scans for AUG. Once it gets there, tRNA can come in with MET or FMET, and then a large subunit sits down. And then, boom, we keep going. See, yes. And then here we go. We're going to do this one as the iPad genetic code sample exam question. And you are going to try to make this happen. So we have a complete sequence of a eukaryotic gene, this whole thing. And you're trying to make this protein specifically. Okay, everything you see is on the template strand is transcribed into RNA. So first tell me which is the template, right? Non-template, hint, has ATG 5' to 3'. You might have to process it. Ooh, I'm guessing you're going to have to do a little splicing, right? How do you know where to splice? Well, you have to end up with this specific protein. And then tell me the 5' to 3' direction of, right, this mRNA. So you're going to need to rewrite the mRNA. So rewrite. You rewrite primary transcript first. Then splice that bad boy. Then don't forget, add cap. You can draw it. And add tail. You can just make it a couple A's for this guy. You can make your hat. However you want to show that. And then rewrite your mRNA. Completely process so that what it makes when it goes to translation, if you stick it through translation, you're going to get MET, ARG, VAL, TIR, ALA. Okay, that's what you have to get. If you get to here and you don't get that, you made a mistake somewhere. Check your splice. And that's it. Thanks so much.